Before we go to the main topic, which is finishing up the Q&A that we did last time, I'd like to remind you that your cystic fibrosis projects need to be attended to. I know that a lot of you are working on them. You need to, for those of you who haven't already told me what time period they'd like for their preview, let me know. It's not urgent yet, but soon it will be. About half of you have reserved a time slot, and the rest of you need to.
Can I ask
Professor?
another question? Oh,
Go ahead.
I
Yeah.
also saw that you just posted something on how to deal with the semester. I was just wondering what that was about. It's from 1:30 to 2:30.
All I know about it is what you saw in that announcement. It looks like it's something like a webinar, and whatever it says on that, it looks like it's trying to help you deal with the stress of doing another semester online. But the only way that we can find out is if you join it and see. That came to me with the request that I distributed to my section.
Professor,
Oh,
keep going.
do we get any extra credit or points for attending?
No, that's extracurricular.
Okie dokie. I'm sorry, Bouchley. You're good.
That's okay. So I have a question on the announcement. I think you sent the announcement last week saying that it's gonna be a quiz 8 today, right?
No, there's no quiz today.
Oh, okay. Thank you.
Not sure when the next quiz will be. I'll when I decide, I will let you know.
Okay.
I won't spring any quizzes on you unannounced. Can you see the worksheet areas of greatest interest?
Yes.
Okay. That's where we need to be. So I'd like to remind you what we were doing last time. If you recall, you submitted questions about things that you either didn't understand or wanted to see explained in more depth. So I compiled your various questions. Many of them are very good, into the categories you see here: energy enzymes, cell division, Mendelian inheritance, photosynthesis, oxidation reduction. We got through the Mendelian inheritance part on Monday. So today I'd like to pick up with photosynthesis and go into some detail and go slowly so that, and a bit reiteratively, so that this complicated material is more easy to understand. But before starting with photosynthesis, I want to make another point. Now, this came about because on Monday, Ben asked me the following question: why are there polar bodies? And neither me nor any of you knew the answer. I'm still not sure I do, but it did cause me to look into it and gain a better understanding of the really significant difference between making sperm and making eggs. You can see that here on this diagram. On the left, that's how sperm are made. You start out with a diploid progenitor cell, which undergoes the first meiotic division and then proceeds straight to the second meiotic division. And that results in the production of 4 haploid cells. And each of those 4 cells is a functional sperm. So from one diploid cell, you end up with 4 functional gametes, sperm. Now, if we look at the same thing in the female, again, you start with what's called an oogonium, but it's a diploid cell, which undergoes the first meiotic division. And you end up from that division with 2 cells, but they're not the same. One is called a secondary oocyte, and it's a normal product of meiosis. The other is called a polar body, and it's not normal at all. It has very little cytoplasm and only just like a little bag of chromosomes, and it dies. Well, not immediately, but eventually it does. And then the second meiotic division comes along, and you get 2 more products of meiosis. Again, one of them is a normal cell, and the other is another polar body. And this so-called ootid goes on to produce the egg, the ovum. So starting with 1 cell, 1 diploid cell, undergo 2 meiotic divisions, but you only end up with 1 viable egg. So how does this come about? Well, I've got 2 diagrams here, and let me go over them in a little bit of detail. So why are there polar bodies? The shorter answer is to avoid twins. On the left-hand side, you see we start out with an oogonium, which is diploid. This is in the female. And when the female is still a fetus, many oogonia are produced, thousands of them, but they're all diploid. Then at puberty, meiosis 1 occurs. At puberty, these oogonia start being released one at a time, 1 every month. And upon release, meiosis 1 occurs. And after meiosis 1, you end up with 2 haploid cells. One is called a secondary oocyte. It's a normal product of meiosis. And the other cell is a polar body, which is not normal. Now, if there happen to be sperm in the vicinity, and a sperm fertilizes this secondary oocyte, it prompts it to go ahead and do meiosis 2. And when meiosis 2 occurs, again, you get a normal cell and another polar body. The first polar body can undergo meiosis 2 as well to produce 2 more polar bodies, all 3 of which degenerate. They don't go anywhere. So what you end up with after meiosis II is what's called a mature ovum that actually has two nuclei. It has the nucleus from the female and the nucleus from the male, which entered when the sperm fertilized after meiosis I. Then these two nuclei can fuse. They're both haploid now. They can fuse and produce a diploid zygote.
Can I ask a question on that, Professor?
Sure.
So, my original question was, why do the polar bodies die in the female? And, what was their purpose? And you just answered that saying that it prevents twins.
Yeah.
But what if you have twins? Wouldn't the polar bodies be living?
Okay. Yeah, that prompts me to back up and say that in some cases, on rare events, this polar body after meiosis I is not a polar body. It's a full-fledged secondary oocyte. And again, if sperm are present, it gets fertilized too, also. And if it does, you now have twins. So if you look at this evolutionarily, over thousands of years, hundreds of thousands of years, and many, many human generations, has there been selection against twins? In other words, are human babies so precious that all of their care and resources of the parents should go into one baby rather than two? I don't know, I'm just speculating, but it's possible that polar bodies are there, that the reason for polar bodies is to prevent twinning. There's more than one way to have twins. One is if the polar body doesn't degenerate and it's fertilized. Another is if there are two different eggs, two different oogonia released on the monthly cycle, and they both get fertilized. And another is that once the zygote is formed, something happens that causes it to split into two, and both cells get a full complement of chromosomes, so you have identical twins. If a polar body got fertilized, these would not be identical twins, because remember, products of meiosis are never like each other and they're never like the parent. So they would be fraternal twins. Now on the right, I made a flow diagram, tried to simplify what I've just gone through here. Point one, girls are born with thousands of primary oocytes. That's these guys, which are diploid. But they're destined to be sex cells. At puberty, a female will start to release one of these primary oocytes every month. And upon the release, it's still diploid. But the release itself prompts it to undergo the first meiotic division, meiosis I. The result of that is this secondary oocyte, which you see here, and a polar body. But the completion of meiosis I makes the secondary oocyte susceptible to being fertilized. If there are no sperm present, it just eventually degenerates. But if there are sperm, it can be fertilized. And if it is, that prompts it, fertilization prompts this secondary oocyte to undergo meiosis II. And now you have a cell with two nuclei, one of which has gone through both meiotic divisions, that's the male nucleus, and the other has gone through meiosis 1, but not yet meiosis 2. But then it does go through meiosis 2, and that produces an egg and another polar body. So the egg is this mature ovum, which now has two nuclei, one from the male, one from the female. They've both gone through both meiotic divisions, so they're both gametes. And now the nuclei can fuse into and become diploid, which is a zygote. And it will then start cell division and become a full-blown individual. So why am I making such a big deal about this? Well, for a couple of reasons. One is that Ben asked, and the other is that it strikes me that we really haven't spent enough time contrasting males and females when it comes to production of gametes. Males start with a diploid and end up with 4 haploid cells. Females start with a diploid and end up with 1 haploid cell. And it does this by this rather convoluted and detailed process that you see illustrated here. So does everybody understand this? Everybody see the distinction and see how it happens in females? It's much more complicated in females. Okay, if everybody's got it down, then we'll go on to photosynthesis. A couple of people asked, can animals undergo photosynthesis? As far as I can tell, the answer is no, they can't, but they can try. So 2 examples here. One is the sea slug, the other is a spotted salamander. Here you see a picture of a sea slug sitting on a bed of algae, which is the food that the slug eats. But it then integrates, it takes chloroplasts out of the algal cells. Algae are green, they're photosynthetic. They have normal type chloroplasts. And the slug integrates these into its own cells. And the presence of the chloroplasts in the slug cells allow them to live up to 9 months without eating anything. They can essentially take advantage of, they have to have some sunlight, but they can take advantage of the sunlight to make photosynthetic products and live off those. So it's like being photosynthetic, but they really aren't. They just borrow chloroplasts from their diet. The spotted salamander is somewhat different. It has what appears to be maybe even a symbiotic relationship, again with algal cells. Here's a picture of a spotted salamander with these yellow spots. So the algal chloroplasts are taken into the salamander cells and sit near the salamander mitochondria. So this is where the symbiosis seems to happen, that the mitochondria use oxygen and carbohydrates produced from the chloroplasts to generate energy for the salamander. And these chloroplasts persist in the salamander throughout its life cycle. In fact, it seems as though they can even go through meiosis, the chloroplasts. And it's been observed that the salamanders seem to do better in light than in darkness. But the point is, they're not dependent on chloroplasts for their existence. They have mouths and they can eat their normal diet in order to get energy. So for those of you who are asking, do animals have photosynthesis? I think it's fairly safe to say no, they don't, but they sometimes interact intimately with photosynthetic organisms. So let's focus on photosynthesis then in plants. Where in a plant does photosynthesis occur? The general answer to that is any part of the plant that's green. So, for example, stems are green. They can photosynthesize. Petioles are green. They can too. But most of the photosynthesis occurs in the leaf blade. And if you look at a cross section of a leaf, various different tissues, there's an epidermis on each side, stomata, and then the mesophyll cells in between. And these are the ones that do most of the photosynthesis. And if you take just one of these cells and look at it more closely, you can see that it has a cell wall, it's a plant, a nucleus, a huge vacuole. Remember, plant cells have huge vacuoles to store numbers of things, including water. And then there are the chloroplasts. These are the mesophyll cells. Then if you drill down, yeah?
Professor, I have a question. So are you saying that photosynthesis cannot occur in any part of the plant unless it is the green part, right?
That's right. So roots, for example, are not green and they can't photosynthesize.
Okay, got it. Thank you.
Look a little closer at the chloroplast. Again, here's our mesophyll cell. You take one chloroplast and blow it up. What do you see inside? Well, it has an outer membrane and an inner membrane and stroma and grana. What are they? Well, the grana are these stacks of membrane-bound vesicles called thylakoids. Well, a thylakoid has a membrane and an internal compartment. The whole thing is called a thylakoid. And a stack of them is called grana. The singular, one member of this stack would be called a granum. And this is where the light reaction occurs. We'll talk more about light and dark in a bit. But the chloroplast has two major reactions, one that occurs only in light, and the other that occurs anytime in both light and dark, but it's called the dark reaction. And that's where we have the Calvin cycle. So electron transport, many of you asked about that in chemiosmosis, occur in these thylakoid membranes. And we'll focus on that up close in just a little bit. So any questions about this? The light reaction occurs in the grana. The dark reaction occurs in the space between the grana, which is called the stroma. So that's where the dark reaction occurs, which means that it can go on in both light and dark. The light reaction can only go on in the light.
So Professor, is the light section the one that's called the Krebs cycle, or is that separate?
That's something different.
Okay.
Yeah, but it's something to keep in mind as we go along, that what we're talking about here is just photosynthesis. And when we get to the Krebs cycle, what it does is take the energy that's generated by photosynthesis and use it, it takes it as food and
Okay.
extracts energy from it.
Got it. Okay.
Oh, were you?
somebody have their mic on? Just let me take a look.
No, it was me. I was asking a question. They just didn't realize my mic was on when they walked in the room, so I apologize.
Okay. All right. Right, stages in photosynthesis. You make this bigger so you can read what's in the Kelvin cycle. So here we see the light reaction on the left, the dark reaction on the right, and at a very gross level, what happens in the chloroplast is that water, carbon dioxide, goes into the chloroplast, and output is oxygen and sugar. What the light reactions, which occur in these grana in the chloroplast, generate both ATP and electrons, which go into the dark reaction called the Kelvin cycle. And what the Kelvin cycle does is an amazing thing. It takes a gas, carbon dioxide, and converts the carbon in that gas into a solid chemical, namely sugar. So if we read the statements here, the Kelvin cycle is a set of chemical reactions that convert carbon dioxide to glucose. It uses ATP and NADPH from the light reaction to produce sugar in a stepwise process. The Kelvin cycle is necessary to capture energy. Without the Kelvin cycle, a one-step reaction from carbon dioxide to sugar would lose most of the energy to heat. So taking all these steps, which you see in the Kelvin cycle, is how the energy is conserved and is not all lost to heat. So carbon dioxide comes in as a gas, it's converted to 3 carbon sugars, which eventually are released from the chloroplast, and 6 carbon sugars are made. And the 6 carbon sugars are then the food for the mitochondrion, which is respiration. So we have photosynthesis on one hand, respiration on the other. Photosynthesis makes food, respiration uses that food. So here are several questions that came up repeatedly in what you submitted. What is oxidation reduction? Redox. What is an electron transport chain? What is chemiosmosis? And what do all of them, either individually or together, what do they have to do with ATP production? And why are they all necessary for ATP production? So let's go back to the beginning and review some of the things we've gone over before. And now maybe they'll be a little bit more meaningful to you. What is oxidation reduction? Well, if you recall, a substrate can be reduced, as in A, or oxidized, as in B. The reduced substance is reduced because it has an extra electron. But if it loses that electron to an oxidized state, a compound with oxidized state, now B is reduced and A is oxidized. So oxidation reduction is really the passage of electrons back and forth from one substance to another. And in the process of doing that, energy is generated. And if you remember the mnemonic oil rig stands for oxidation is loss, that you see here. Oxidation is lost, reduced is gain. So this is the basic information about oxidation reduction. The next thing you need to know is that phenomenon, passing electrons back and forth, is the electron transport chain. And what this illustrates is one step, one link in the electron transport chain. Electrons can be moved through molecules called intermediate electron carriers. In plants, it's NADP, the carrier. In animals, it's NAD. So what you see is this step, passage of electrons from reduced to oxidized, it happens and the electrons are passed to a carrier, in this case, NAD, so that it becomes NADH plus a proton. It, in turn, can pass its electron and proton on to the next carrier, in this case, for example, cytochrome. So again, you get an oxidation reduction reaction. And this is called redox, this passage of electrons from one carrier to the next, and that carrier to the next carrier, and so on. And that leads us to an electron transport chain. Electron transport chains occur in membranes. And the membrane can be either in the chloroplast or in the mitochondrion. Regardless of which one it is, you see the membrane here. Within the membrane are these proteins, which are where the electrons are transferred. So in this case, it starts with NADH plus a proton, which are contributed to a flavin, a carrier. Another oxidation reduction reaction occurs, and the electrons go to compound Q, and there they're transferred to cytochromes, several different cytochromes, and then to another protein, and finally, electrons are transferred to oxygen, and that's converted to water. Oxygen plus 2 protons is H2O, water. But now what's happened, as these electrons are passed along from one to the other, energy is being lost. The electrons are becoming less and less energetic. And part of the reason for that is that they're producing protons, which are accumulating on one side of the membrane. That causes a gradient, a proton gradient. So there's many more protons on one side than on the other. And that's what leads to chemiosmosis. The passage of these protons through this channel in the membrane, which contains, among other things, ATP synthase, the energy from these protons goes to convert ADP plus inorganic phosphate to ATP. And this is a very efficient process, as you'll see. You get a lot of ATPs made from one molecule of glucose. So this is the electron transport chain. And I put this in to give you a close-up of the electron transport chain and its coupling to chemiosmosis. Again, this goes on in a membrane. The membrane can be either in the chloroplast or the mitochondrion. Again, you start with a reduced compound which transfers its electrons into the electron transport chain where they go bouncing along. And as they do, they release energy in the form of protons accumulating on one side of the membrane. then when they diffuse through the membrane, through a channel that contains ATP synthase, the energy provided by these protons makes ATP out of ADP and phosphate. I'd like to show you a video illustrating this. And I think to do that, can you see the video? I don't think so. I have to stop sharing this screen and share another one.
After glycolysis, the link reaction and the Krebs cycle have taken place, the reduced form of the electron carriers, that is NADH and FADH2, are sources of electrons for the electron transport chain. These are transported to the inner membrane of the mitochondrion and to the cristae. In the inner membrane, there is a series of electron carriers represented by the white circles here. On the inner membrane, NADH is first oxidized to NAD+, releasing 2 electrons, represented by E- here. Those electrons are picked up by the first electron carrier in the chain. Those electrons are then passed down a series of electron carriers found in the inner membrane, each successive carrier having a slightly higher electronegativity, or that is, a stronger attraction for the electrons. As this happens, energy is released, and this energy is used to pump protons or hydrogen ions via active transport into the intermembrane space. FADH2 enters the electron transport chain at a lower energy level than did NADH. The FADH2 is also oxidized into FAD, releasing electrons which are picked up by the electron carriers in the chain. These electrons continue to pass down the series of electron carriers, and with each successive carrier release energy. This energy continues to be used to pump protons or hydrogen ions from the mitochondrial matrix into the intermembrane space. At the very end of the chain, the final electron acceptor is oxygen. 2 de-energized electrons combine with 2 hydrogen ions and the oxygen in order to form water, one of the products of aerobic respiration. As these electrons continue to be passed down the electron transport chain, hydrogen ions or protons continue to be actively transported from the mitochondrial matrix to the intermembrane space, creating a proton gradient, where the concentration of protons is higher in the intermembrane space than it is in the matrix. Therefore, the hydrogen ions diffuse passively through a channel in ATP synthase back into the mitochondrial matrix from an area of high concentration to an area of low concentration. The enzyme harnesses the available energy, therefore allowing for the phosphorylation of ADP into ATP. This process is known as chemiosmosis. For every 1 molecule of glucose, a total of 32 ATP are produced through the electron transport chain and chemiosmosis. So as a review of aerobic respiration, if you recall in glycolysis, we used 2 ATP but then produced 4, so there's a net gain in ATP of 2 ATP. The Krebs cycle produced 2 ATP, and the electron transport chain and chemiosmosis produced 32 ATP, and this is all for 1 molecule of glucose. Therefore, in total, for 1 molecule of glucose in aerobic respiration, we produce 36 ATP. So overall, aerobic respiration can be represented in an equation, which would be C6H12O6, otherwise known as glucose, plus 6O2 produces 6 CO2 and 6 H2O, along with energy in the form of ATP or lost as heat.
Are we back? Can you see the chloroplast chemiosmosis?
Yeah, we're good.
Okay. All right, so we just watched this video. Any questions or comments about the video and how it relates to the diagrams that I've shown you? So what the video was showing was the electron transport chain within, the video was focused on a mitochondrial membrane, but the same thing happens, or very similar thing happens in chloroplast thylakoid membrane. Was it clear to everyone?
So the protons are transferred from the original side across and then back. That's what.
They start at the left and they're transferred
Or just some on the other
towards the
side.
right. Yeah, and then the final acceptor for electrons is oxygen. And so you have electrons or protons, hydrogen ions, combined with oxygen, 2 for 1, you get H2O. 2H plus, a half O2 gives 1 molecule of water. So that's the end. The end of the electrons is in water.
The de-electrified or whatever.
What?
The less electrified electrons or whatever.
Right, they've lost most of their energy by the time they get that far. And when we talk about photophosphorylation, you can see that they need light to maintain their energy. If they don't have light in the mitochondria, then they lose it eventually to water. Okay, so let's talk about electron flow and light reactions. So now we sort of transitioned into mitochondria. Now we're back to chloroplasts. Chloroplasts have 2 what are called photosystems, photosystem 1 and photosystem 2. In terms of electron flow, they're actually backward. Electrons start in photosystem 2 and end up in photosystem 1. So in a chloroplast, a photon of light comes in from the sun and is absorbed by one of the chlorophyll pigments, in this case, one that absorbs at 680 nanometers. And when it does, a couple of things happen. First, a water molecule is split into oxygen and 2 hydrogen atoms, hydrogen ions, H plus, and their corresponding electrons. Remember, a hydrogen atom is a proton plus an electron. The hydrogens stay here. The electrons are highly energized by this photon of light. And that indicates by the arrow going up to a higher energy level. And that's where it enters the electron transport chain. Now, this is very similar to the electron transport chain that you see in mitochondria, but now it's occurring in a thylakoid membrane instead of in the inner mitochondrial membrane. And when this happens, there is a substrate level phosphorylation. So a little bit of ATP is made from this electron transport chain. And in going through that and in generating this ATP, the electron loses energy. And now it enters photosystem 2, which is a different chlorophyll pigment, absorbing at 700 nanometers. But now another photon of light comes from the sun and re-energizes this electron up to a high energy state again, so that it can be passed on to more electron carriers, a flavin, and finally to NADP, which is converted, which goes from NADP to NADPH, which is an energy carrying molecule, plus a proton. So this all happens in the light reaction. The Kelvin cycle, you're staying in the protoplasm and the chloroplast. The Kelvin cycle is next. So what does the Kelvin cycle get from the light reaction? It gets ATP and NADPH. So just to review in a different format, here you see a granum. This is a thylakoid membrane with these electron carrier proteins in the membrane. Photons of light enter, and you get an electron transport chain going, which ends up releasing ATP and NADPH, which go to the dark reaction, the Kelvin cycle. So this is a thylakoid compartment, this part in here. The membrane is this phospholipid bilayer. And then the Kelvin cycle occurs in the stroma. Still in the chloroplast, but now it's in the stroma, which is the space between grana. Okay, this is a reiteration of that point that ATP is produced by a process called chemiosmosis. And again, you see the lipid bilayer, see the light energy coming in, activating electrons in photosystem 2 and photosystem 1. The electrons go through an electron transport chain. And as they do, so in the video, when this happens, protons are pumped across the membrane from outside to inside. And that makes a gradient. So when the gradient is formed, or after it's formed, the protons can pass back through it, through this channel, which has ATP synthase. This is chemiosmosis. And this is the highly efficient process of generating ATP. These little blue dots and red triangle or rectangle, you've seen before, and I put that in just to remind you what chemiosmosis is. It's the passive transfer of an ion, in this case a proton, from a high concentration across the membrane to a low concentration. And it's this passage through, in combination with the enzyme ATP synthase, that generates ATP. So now let's take a little closer look at the Kelvin cycle, which is the dark reaction of photosynthesis. The input comes from the light reaction. This little inset here shows the light reaction, which generates ATP and NADPH. So these are used in order to convert a gas, carbon dioxide, into a solid. So for example, in this step, 6 ATPs are used. And in this step, 6 NADPHs are used to convert one intermediate to the next. And when and let me emphasize that This first step, it's called carbon fixation. It's catalyzed by an enzyme known as Rubisco, which does an amazing thing. It takes a gas and makes a solid out of it. So it takes 3 molecules of carbon dioxide, takes the 3 carbons from that, and makes a 3-carbon intermediate. I have here the tertiary structure of Rubisco, just to show you that the active site of Rubisco actually binds a gas. It actually binds carbon dioxide. And then as a result of its catalytic action, it converts the carbon dioxide into a 3-carbon solid. And partway through the Calvin cycle, glyceraldehyde 3-phosphate, one of the intermediates, leaves the cycle, and 2 of those molecules, and this is the structure of glyceraldehyde 3-phosphate, 2 of these 3-carbon molecules are stitched together to make 1 molecule of glucose, which has 6 carbons. And it's glucose then that is food for mitochondria. So this has all happened in the chloroplast. Glucose then undergoes glycolysis, and that's what the mitochondria use in order to provide energy from food. And that brings us to talking about respiration. It's really hard to talk about photosynthesis without also talking about respiration, because many of the same processes are involved in both. Now, the next slide set, which looks like it's going to be next Monday, we'll talk about respiration in particular. But until we get there, here's a first look. And now remember, we're moving from the chloroplast to the mitochondrion. Respiration occurs only in the mitochondrion. We have, within the cell, we have the cytoplasm and the mitochondrion. Glycolysis occurs in the cytoplasm. Remember, we're getting glucose from the chloroplast. Glucose is converted to pyruvate, so it's a 6-carbon sugar to a 3-carbon sugar, and it's pyruvate that enters the Krebs cycle. Not only that, but the process of glycolysis has a substrate level phosphorylation, generating ATP, and it releases electrons carried by NADH. And where do they go? To the electron transport chain and oxidative phosphorylation, and which involves chemiosmosis. And that's where you see this star of burst of ATP is larger, because oxidative phosphorylation produces a lot of ATP compared to substrate level phosphorylation, which only generates 1 or 2 molecules per glucose. So glycolysis has substrate level. The Krebs cycle has 1 substrate level. And as it turns, it keeps churning out 1 or 2 molecules at a time. But what's more important from the Krebs cycle is that it generates NADPH, or NADH in this case, which can enter the electron transport chain and make a lot of ATP. So this glycolysis, let's look a little more closely at it. Glycolysis takes a molecule of glucose, 6-carbon sugar, and converts it to 2 molecules of pyruvate, each of which has 3 carbons. And in doing so, it generates 2 ATP molecules and 2 NADH molecules. And both of these are used by the mitochondria. Usually glycolysis is shown as a single step, glucose to pyruvate. In fact, it's 9 steps. And I don't expect you to memorize all of these 9 steps, it's just a bunch of intermediates. But I want you to know that glycolysis is not a simple one-step process. It's a very complicated for a variety of other reasons. And that's the only reason I show you this diagram. And now let's look more closely at the Krebs cycle. So here's a cell. You see here is its nucleus. The little object is a mitochondrion. You blow it up, you can see that it has this internal membrane structure. And if you look at that closely, you can see that the mitochondrion has an outer membrane, an inner membrane. This highly convoluted structure is the inner membrane, and then a space between them. And the space between them is very important. So glycolysis occurs in the cytoplasm, and glucose is converted to pyruvate. Pyruvate passes across the mitochondrial membrane, both membranes, and is converted to acetyl-CoA. The acetyl-CoA is the first step in the Krebs cycle. The Krebs cycle is the same thing as the TCA cycle. It stands for tricarboxylic acid cycle. But think, when you see that, think Krebs cycle. So as the Krebs cycle turns, it's releasing carbon dioxide. That's part of respiration. When we breathe, we take in oxygen because we need that for respiration, and we breathe out carbon dioxide. And the reason we do is because of the Krebs cycle. The most important thing the Krebs cycle does, it does have a substrate level phosphorylation, but it generates reduced electron carriers. And these reduced electron carriers go into the electron transport chain. This is what you saw in the video. They come in from the matrix into the membrane and are passed along from one carrier to another, and finally come out after they've lost their energy, come out and combine with oxygen to form water. So the other product of respiration, besides carbon dioxide, is water. While they're passing through these electron carriers, they're pumping protons from the matrix into the intermembrane space. So you end up with a high concentration of protons, which can then passively go back out through the membrane into the matrix. And when they do that, they release energy. And that energy is used to make ATP out of ADP and PI. And this is a much more efficient process than substrate level. So this is called oxidative phosphorylation. What you see here in this inner mitochondrial membrane, that is oxidative phosphorylation, which is an electron transport chain coupled with chemiosmosis. Chemiosmosis is represented by these purple bundles here. ATP synthesis. Any questions? Key roles of acetyl-CoA. A couple of you asked about acetyl-CoA and how it can be both catabolic and anabolic. Well, in the Krebs cycle, It's catabolic. It tears things, it contributes to tearing things apart and generating energy. So again, you don't need to know all the names of these intermediates in the Krebs cycle, but you do need to know that it generates NADH and flavin FADH2 and a little bit of ATP. So the Krebs cycle itself is not oxidative phosphorylation, it's substrate level phosphorylation, but it provides the grist for oxidative phosphorylation. It makes a little bit of ATP, but mostly it makes NADH and FADH2, which go into the electron transport chain in the membrane. And that's what generates the bulk of the ATP. Acetyl-CoAX is a crossroads compound. You'll notice here in the Krebs cycle, it's the first step. Krebs cycle starts with acetyl-CoA. And that's involved in this catabolic process, but it can also be used as a building block for fatty acids. And in that sense, it's anabolic, builds things up. And so I want you to focus here just on the acetyl-CoA in the middle of this diagram, and notice that it can lead to fatty acid synthesis. Shown here, acetyl-CoA through an intermediate to fatty acids, represented by these chains of circles. And to remind you what fatty acids really are, long hydrocarbon chains with a carboxylic acid at one end. C double bond O, OH. That's what makes a fatty acid an acid. And remember that fatty acids can be either saturated, where there are no double bonds, or unsaturated, where there can be one or more double bonds. And I emphasize this because of the importance of fatty acids in membranes. Membranes are essential for all the processes that we've been talking about. So if you just look at some of the diagrams that we've had, here's a membrane. What is it? It's a lipid bilayer. If we go to photosynthesis, it's a membrane, another membrane in the thylakoid, electron transport and photophosphorylation. It occurs in the thylakoid membrane. And these membranes are largely, well, the basis of all membranes are these phospholipid bilayers. So you remember a phosphate group can get attached to this hydrophilic end to make it into a phospholipid, which is, if you remember the word, amphipathic. It means its one end is soluble in oil, the other end is soluble in water. Okay, we're almost out of time. And that's all I have to say for today. Any questions? Has this been confusing or helpful?
Our request, Professor.
Yeah.
Can we please, I don't know what other students are in this boat, but could we have an extension for the worksheet that was due last night? I'm just like, it's really like difficult.
Yeah. Take the rest of the week to finish that.
Okay, thank you. Cause yeah, I'm working on it. It's just like, I don't know. I feel like it's a doozy.
Yeah, okay. Yeah, I'll give you to the weekend.
Thank you very much.
Mhm. Okay, anything else?
Professor, I have a question on the mutations, the group project. Can I use the sources other than the one that you assign on the
Oh, yeah, you're expected to. The links that are in that document are very limited. So yeah, you should explore the web. In most cases, you'll need to go into the National Library of Medicine and look up some primary literature.
Okay. Thank you.
Okay, anything else before we go? All right, well, I will end the session then.
Thank you, Professor.
Okay,
Thank you.
yeah, all right.
Thank you.
Gonna stop sharing and end the session. I'll see you all on Monday.
See you Monday, Professor.
Bye.